Well, the next speaker is ISB's very own Allison Kudla, and Allison is really a marvelous representative of ISB. She went to the University of Washington. She has committed a lot of her career thinking about how you could bring systems thinking to paintings. She's had exhibitions in the New World, in the Old World, and at some very famous places. We're terribly proud of her and wondering just what she's going to tell us about now. So, Allison, it's a pleasure to have you here.
Yeah, maybe it's better. Then I can. Yeah. Okay. Can you hear me? Yes. Okay. How about that?
Yes.
Okay, great. So, as Dr. had mentioned, my name is Allison Kudla, and I am the communications designer here at ISB. And I am going to talk today about biological systems art, which is a type of art that I began to define while I was working on my PhD at the University of Washington in their DX Arts program. And we'll hear more about DX Arts, I'm sure, from others, but it does stand for the Center for Experimental Arts, the Center for Experimental Media and Digital Arts. Before that, I was at the School of the Art Institute of Chicago, where I received a BFA, DX Arts, Center for Digital Arts Media at UW, and then I also spent some time at the Srishti School of Art Design and Technology in Bangalore, India, where I found a really cool collaboration with their National Center for Biological Sciences, where I was able to work on biological art projects at their NCBS in Bangalore. So I have my presentation set up in 3 sections. The first section kind of talks about the framework or the 2 main hallmarks that get me to understanding this concept of biological systems art. And then the second section, I will explain what is biological systems art. And in the final section, I will use one of my artworks to help explain what biological systems art is. So moving forward. So this first hallmark I really got to learn when I was getting my undergraduate degree at the Art Institute of Chicago. Basically in art, matter and meaning are in a constant feedback loop. And what is really remarkable about the School of the Art Institute of Chicago is that you don't have to pick a major. You are required to take a certain number of practice-based studio classes and a certain number of art history, theory, and liberal arts classes. But what those classes are, what medium you choose to make work about, with and about, is kind of up to you. So you can mix and match and move around fluidly in an interdisciplinary way. So I got to move from painting to sculpture to fiber to video, and really what was driving me were my ideas, but at the same time, I'm learning in the process that materials and technologies can also really drive artistic ideas. And as well, artistic ideas grow new technologies and materials. So really, we're never not thinking about material and meaning as anything other than being coupled. So that is kind of, it's this exploration of where imagination meets material, where the creative process begins. So I hope that's making sense to everybody. And so the kind of takeaway from Hallmark 1 is really choose the right medium for your idea or your message, and then also be aware of and cultivate your medium's influence on your work's intended experience. So understand your materials. Next hallmark, so when I was, like I mentioned, I moved from painting to fiber to video. video. Finally, my last couple of years at the Art Institute of Chicago, I was doing basically what is called new media art. So they had an art and tech department. And I was doing visual programming. So I was essentially creating animations that were nonlinear. So these animations would respond to the external environment. So I would have a sensor that would maybe be picking up audio levels, capacitance, and then it would be altering something happening on a screen. And so I was already starting to understand this concept of systems art simply by working with computers and by working with new media. I had an input, an output, and a logic system. I was moving away from an object like a painting or a sculpture and moving towards art that is more transactional, dynamic, art's a system. And of course, this type of art incorporates engineering and computation and that's going to be annoying. Right, so operating system. Okay, right, so at the time I'm making animations and they are definitely residing on a computer's operating system. So then comes the time for me to move on to a PhD. And if anyone in this room has gone towards a PhD program, then you know what happens when you start a PhD is that you question everything and you become inspired and you start to create your own research trajectory. And that's exactly what happened at my very first conversation with my committee chair and advisor, Sean Brixey. He explained to me this very interesting concept that he had been cultivating. And this concept is called emulation. Now, it is a very difficult concept to explain, so I will show you a quote. And I will read it. The paradigm shift that emulation art suggests is the inevitable result of hybrid art research practice at the intersections of scientific discovery, informatics, and aesthetics, as we seek to understand the universe as an operating system in which we perpetually engage on both a microcosmic and macrocosmic level. Now that's quite a mouthful. What I take from that, it's very directly related to systems art, systems biology, looking at things from all scales and also recognizing that the physical universe is an operating system. So back to Hallmark 2, but now we've got the word emulation there. So works of art need not be confined to traditional materials, maybe not even confined to a computer's operating system. The operating system for art can be the physical universe. That opens it up quite a bit. And now, art always exists in both time and space. We would never consider art as not existing in both time and space. And then finally, once that work of art has been created and it's experienced, the process of the audience receiving that work becomes part of the system. So there's a lot to think about once we start thinking about systems in art. It's no longer an object. So, I want to go back to this screen. So here we see Sean. This is Sean Brixey, my committee chair at the University of Washington, and he is working on, right here, he's at his project Voltar, where he is cloning glaciers. So he's gone up to the northern hemisphere and he's taken samples of receding glaciers and figured out how to clone them. So Sean works with physical mediums. So he's interested in water and light. But I have always been very fascinated by biological organisms. And I wanted to figure out, how can I take this emulative approach and bring it to a biological emphasis? So how are biological systems different than physical systems? How would what I would be doing be different than what Sean is doing? What are the behavioral aesthetics and algorithms that are embedded in living systems? How can I unravel the mysteries to create poetic experiences and, in doing so, new material forms? So I basically decided to kind of come up with a parallel track that biological systems art is running on to computational art, since we can kind of understand a computer operating system. So once we take that language and then reference it to biological systems art, we can kind of start to understand the definition I'm creating here. So biological systems art are very similar to algorithmic computational or simulative art, But instead of expressing the modulation and output of algorithms with a digital computer, say a display, the output is emulative because it's expressed in genes, cells, and organisms. Further, because you're working with living systems, the time cycles have nothing to do with, well, I shouldn't say nothing to do, because sometimes these are interdisciplinary systems, but time is not only about how fast can your computer run or what is the frame rate of your movie, but rather you're constrained by biophysical time, the amount of time it takes for a biological system to evolve, grow, express, behave, senesce. And of course we can begin to consider mutating organisms to change their time cycles. But again, that would be a bit like figuring out how to overclock a plant, which I will show you later has been done. So, further, embedded within biological systems is the poetry and dialogue of the predictive versus the emergent. Also, state transitions, and of course, life and death. So if you're working with living materials, you are inherently going to be dealing with these concepts. Finally, going back to this concept of the predictive versus the emergent, biological systems are repetitive in many ways, and their mechanics can be studied, but they are highly complex. So in biological systems art, I put the biological systems into a process of recursion, a pattern, to create a platform for experiencing fascinating variability. So by trying to create repetition, I then see variability. And I'll show that in a final work later. So again, if you are working on biological systems art, there is something called, I've come up with this idea of a behavioral aesthetic. So what are the metaphors that are inherent in biological systems? Say you want to make work with a biological system. What are the kind of, metaphors you're going to be dealing with? So here's a list of some: differentiation, genetics, communication, signaling, response, circulation, circadian rhythms, reproductive processes, symbiotic relationships, the process of death. All of these metaphors become a story or a poetic framework for what you're doing with your artwork or the system that you're creating. So I chose to focus on differentiation, circadian rhythms, communication in the works that I did. So the first project that I made at DX Arts that really focused on biological systems art is a project called The Search for Luminosity. What we see here is the oxalis plant, and if you're familiar with this plant, it is phototropic, meaning that when it's, it basically lifts its leaves in the presence of sunlight. So it has this very dramatic gesture of lifting its leaves in the sun. And I first saw this plant and thought, well, that's cool, I like how it moves, I wonder what I could do with that. And then I looked at it more and discovered that it actually will open its leaves in darkness. And I thought, that's interesting. So it's for biochemical reasons that even in darkness it will lift its leaves, because it's kind of expecting to get sunlight. It's ready for it. And so there is kind of a memory, a sort of biological memory of the day before embedded in these plants. And so I thought, great, I can use technology to reverse the hierarchy of physical and biological. So I created a sensor that would watch the plant, wait for it to wake up, and then once it saw it lift its leaves, it would turn its light on. So I effectively gave the plant the ability to communicate with the acting sun in its universe. And so it becomes a very small poetic framing of the impact that biological systems can and do have on the physical world. After I finished that project, I got to meet Dr. Liz Van Volkenberg, who works at the University of Washington's Department of Biology, and she works in plant neurobiology and plant physiology, plant signaling and response. And I was very grateful to get to have many amazing conversations with her. And it is a very fascinating topic, and I did interview her for my dissertation, and this is a quote from that dissertation. But essentially, in plant neurobiology, or this concept of plant signaling and response, there's this idea of saying, well, a plant has a behavior or it stores chemical information, but what causes objection is once the word cognition is applied. So values become charged when people want to stand up against the use of this term to apply to a plant. And I think it's just really fascinating that this is kind of a line that we have. And I won't go into it too much further, but if you're interested in this concept, Liz is here today, so you could talk to her. The next work that I made was this one called Capacity for Urban Human Error. I was learning about 3D printing and fabrication technologies. And I was interested in the idea of a mechanical system being involved in the differentiation of an organism. So I had read about 3D printing organs, and I thought, well, I'm naive enough to take this on. I can do this. I'm going to try to figure out how to 3D print a plant. So I went ahead and built this XYZ plotter, figured out how to extrude material from it, and I began extruding undifferentiated plant cells. And the idea was that the form would come from the computer system. Well, undifferentiated plant cells are pretty difficult to keep happy. They like to have sterile conditions. So, Oh yeah, good call. Thanks, Callie. You're awesome. I do have a video, but in the interest of time, I'm going to move on. And in the last section, I'm just going to talk about growth pattern. So the premise for growth pattern is the merging of a living botanical system with a cultural legacy of botanical motifs. So again, there's some poetic framing here. What I'm doing is attempting to structure a living organism inside of an abstraction of itself, thus causing a framing of control and plasticity to unfold in time. So what is this project? Well, it starts from my sense of awe and wonder when I found out that plant cells are totipotent. What is totipotent? Well, it means that any cell in a plant can become any organ in a plant. So that means that depending on the ratios of auxins to cytokinins, which are essentially plant growth regulators or plant hormones, cells have the capacity to differentiate into any organ. Through chemical intervention, functional formulas embedded in plant cells allow for this quality to be manipulated. So I've laid out these three, it's a very simple, kind of like a toggle switch here. It was mind-blowing to learn this. You switch this one up, the leaves grow, you switch this one down, the roots grow, and you keep them the same and it's undifferentiated. And so this formula really begins to unlock one aspect of the operating system of the plant. And so as a non-scientist learning this, I was overwhelmed. I decided to go with this formula and take leaf cuttings and grow new leaves. How do you take leaf cuttings and grow new leaves? Well, here is a time-lapse video of a leaf disc that over time its edges have kind of cauterized, callus tissues being created, and new shoots are growing from it. So this is a time-lapse video, an aerial view of the petri dish. Those are condensation droplets. I hope you can see the shoots. They're coming out. It's very easy to see. And so I wanted to make an interesting pattern of these, and I wanted that pattern to have a relationship to my material. And so I studied botanical motifs, and I found that bilaterally symmetrical and radial patterns were a common way of representing botanical forms, which works perfectly with the leaf itself. So as you can see, I very methodically cut this plant tissue. I then had to sterilize this plant tissue. It involved putting it through alcohol, bleach, soap, sterile water. It's a very intensive process. And 3 states were the outcome. So in the kind of perfect balance state, sterility is achieved. No microorganisms grow, and new sprouts appear after 3 to 4 weeks. And ideally, for me, it was going to be that all of the tiles are going to grow in this way. And I was really looking forward to seeing what kind of hyper symmetry I was going to get within my beautiful pattern. And I wanted to understand what is the reason why shoots grow where they do. And is it just random? And could I learn something from this? So that was kind of my inquiry. But what I found out was that, well, life happens. And some tissue was not sterilized enough, and hidden fungi and bacteria emerge in a matter of 1 to 2 days sometimes. And that's the second state you see here. What's cool about this particular one is because of the methodical way I cut, it translated the same little bit of fungus from one side of the dish to the other, creating symmetry in the fungus. In the final dish, I was a little overzealous. I cleaned that leaf a little too long. It's very delicate, maybe 30 seconds too long. And the leaves after, a week or so, the chloroplast just kind of fell off because the leaf was dead. And I didn't know. So those were the 3 outcomes. And the work was exhibited as a kind of like a display. So it was on the ground and it was lit from beneath. So it was kind of like walking up to this gigantic screen. But it wasn't a screen because it was a living system with condensation and growing things. And so art historians have since called this a generative display. Now, you might say a generative display is when you write the computer code that draws a picture on a screen that you aren't expecting to see. Well, in this case, it's generative because I don't know what microorganisms ended up in the system that I have created. And it is going to generate something, and it's going to be different every time. But what isn't going to be different every time is the structure or the rules or the composition by which I set out to make it. So occupants get to see the morphological changes all at once over time because there are so many. And so this is the recipe. So this is what I use every time. I can change the pattern, obviously, but here's the recipe. I kind of refine it as I go a little bit, but for the most part, what I'm getting at is the recipe is always the same, the outcome is always different. And because of the recipe, I was able to, in future instantiations of the work, have other people learn how to do it, so that I unfortunately didn't get to fly back to Belgium in this case, but it was great to be able to transfer that knowledge and not have to be there to make the work. And here are just a few pictures before I close. And I want to bring this back to my time at ISB. This is where I see the connection. So, is the plasticity of this living system a window into how humans could manipulate and potentially reverse entropy? So when I learned about the operating system of the plant and the way that I could manipulate these cells to kind of change state, I suddenly realized that's kind of what the really smart scientists I work with are doing at a much greater level, figuring out how to look at state transitions, understand them, and potentially reverse them. And so I'm profoundly grateful to be working here and really grateful to have had the time to share my work with you. Thank you.
So we have time for a question or 2 or 3 if there are any questions. Yes?
I was wondering, Allison, if you are working with the telomeres in relation to the system of entropy and so forth. It would be a fascinating study, I would think, because there's so many things going on now about various things that affect the diminishing of the telomeres.
I am not actively working on that, but would love to. One of the things that I have realized is that to do these kinds of works, you need collaborations, you need partners, just like scientists do. And so to grow my research, that's what I need. Thank you.
Any other questions out there? Okay, thank you again very much.